My research focuses on understanding functional variation in human genomes. Basically, we're all different, we have our unique genomes, and somehow those genetic differences give rise to the human diversity that we see amongst us, which is something that I've always wanted to study. We are trying to understand how genetic variants affect gene expression and other traits of the transcriptome and gene expression patterns. And one of the major motivators for this work is to understand genome-wide association hits. So when we have a genetic locus that has been identified to be associated to some disease, in order to actually develop drugs and interventions and build biological understanding of the etiology of the disease, we need to understand what those genetic variants are actually doing in the cell and in the body. The bread and butter of our research is integrating genome and transcriptome data to understand how genetic variants affect transcriptome traits. When we are studying genetic effects on different traits, we first need to have data of genetic variation, which is of course DNA analysis by SNP arrays or sequencing, and then we need phenotype measurements. But since we are interested in cellular traits, we are using sequencing assays to measure cellular phenotypes such as gene expression. Integrating genome and transcriptome data can happen in many ways. One of the key approaches is association analysis, where we take genetic variants and genotypes from different individuals and associate that to expression levels that have been measured by RNA sequencing. We can also do allelic expression analysis, where we distinguish the expression from each haplotype of a gene in a given individual. And both of these approaches are very powerful and complement each other. I think that studying many different data types in an integrated manner adds tremendous value to research in general, in particular in human genomics. One of the things that I find really exciting in this space of studying functional effects of genetic variants is that even though we are starting to know and understand quite a bit of how the genome works, we are still very, very far from having a full understanding of the functions of the genome and the complexities of the cell and how these things eventually lead to different risks of diseases. There is this tremendous opportunity to make big fundamental discoveries in genomics right now, and it is a huge privilege to be part of this generation of geneticists and biologists.